Hey everyone, and welcome. Today, we're diving into something that sounds like it's straight out of science fiction. What if you could have a single, permanent treatment for thousands of different genetic diseases, all at once? Well, it's starting to look like a very real possibility.
So just to understand the scale of what we're up against, you have to start with this number. There are more than 8,000 known genetic diseases. We're talking about conditions that affect hundreds of millions of people all over the world. It is a monumental medical challenge. And that really brings us to the core question here. What if, instead of trying to fight these diseases one by one, we could find a common enemy, a single thread, and develop one therapy that could work for a huge chunk of them?
You see, the way we approach gene therapy right now is what's called allele-specific. That's a fancy way of saying each therapy is tailor-made to fix one single genetic mistake. And with over 200,000 known disease-causing mutations out there, well, that would mean developing and testing 200,000 different drugs. It's just not going to happen. We absolutely need a new way of thinking about this.
And this right here, this is the heart of that new approach. It turns out that a huge slice of the pie, we're talking nearly a quarter of all the genetic errors that cause disease, they all come from a single type of mistake. It's called a nonsense mutation. And the fact that it's so common makes it the perfect target for a therapy that could work across the board.
So what even is a nonsense mutation? Okay, think of your DNA like a giant recipe book for building all the proteins your body needs to function. A nonsense mutation is like a typo in that recipe that just randomly inserts the word stop right in the middle of the instructions. The cell's machinery just stops reading way too early, the protein never gets finished, and boom, that's what leads to disease.
Now, for a while, scientists have had this really clever idea. What if you could just tell the cell to, ignore that accidental stop typo and just keep reading? It's a brilliant concept, but the solutions we've had so far have some pretty serious flaws.
The tool they've been using for this is a molecule called a suppressor tRNA, or sup-tRNA. You can think of it like a molecular patch. So when the cell's protein-building machine hits that premature stop sign, the sup-tRNA jumps in and says, nope, ignore that, just put this amino acid here instead, and production keeps on going. It's a really smart workaround, but, and this is the key, it's not permanent.
And that leads to a whole host of challenges. Because it's just a temporary patch, you need to take it for your entire life. Its potency can be kind of low, which means you might need really high doses, and that can get toxic. Plus, they're often delivered using viruses. The bottom line is, we need something better. We need something permanent. And that brings us to the hero of this story, a brand new strategy called PERT. Now, this isn't a temporary patch. This is a one-time, permanent genetic rewrite designed to fix the problem at the source for good.
PERT is short for prime editing-mediated read-through of premature termination codons. That's a mouthful, I know, but the key part is prime editing. It's an incredibly powerful gene editing tool that works like a biological find-and-replace function. It lets scientists rewrite the genetic code with pinpoint accuracy.
And the strategy itself is just beautiful in its simplicity. Here's how it works. First, the system finds a spare tRNA gene that the cell doesn't really need. Second, it uses that hyper-precise prime editor to rewrite that gene's code, basically transforming it. And third, the cell itself now starts producing that ignore the typo molecule permanently. In one shot, the cell becomes its own medicine factory.
But here's the thing. To make this work, you can't just use any old ignore the typo molecule. You need the absolute best one, the most effective version you can possibly make. So researchers went on this massive quest to engineer the perfect sub-tRNA.
And when I say massive, I mean it. They didn't just test a few options. They screened all 418 known human tRNAs. And then they created and tested tens of thousands of different versions of them. The goal was to find the one true champion, the single most powerful design possible.
And this chart just shows you how much all that hard work paid off. A simple, basic sup-tRNA had a pretty modest effect, but the fully engineered version they created, it was about 5 times more powerful at getting the cell to read right through that mistaken stop sign. That is a gigantic leap in effectiveness.
Okay, so they've engineered this super potent molecule. It looks great on paper, but the next and most critical question is, does it actually work in a real-world setting? Can it rescue cells from actual human diseases?
And the results from the lab were just incredibly promising. They tested it in cell models for devastating illnesses like Batten disease and Tay-Sachs. A single PERT treatment restored between 17 and 70% of normal enzyme activity.
Now, what's so important about those numbers is that for many of these diseases, that's well within the range you need to see a real life-changing therapeutic benefit. But, the ultimate test is never in a petri dish. It's in a living, breathing organism. And the most powerful proof for PERT came when they tested it in a mouse model of Hurler syndrome, a really severe genetic disorder. This was the moment of truth.
And the results were just stunning. Before the treatment, the mice had all the classic signs of the disease: a toxic buildup of molecules, severe organ damage, the whole thing. But after just a single PERT treatment, the toxic buildup was cleared. The organ damage was resolved. The mice showed a near-complete rescue from the disease. It was a total turnaround.
And here's the truly wild part. To get that near-complete rescue, do you know how much of the enzyme they had to restore? Just 6%. About 6% of the normal activity was enough to achieve that incredible result. That just speaks volumes about how potent this engineered therapy really is.
So, let's step back. What does this all mean for the future of medicine? This is way bigger than just one disease. This is about the promise of a truly disease-agnostic cure, a single platform that could be used for hundreds of different conditions.
Of course, with any technology this powerful, the first question you have to ask is it safe? And the researchers looked at this very, very carefully. What they found was that PERT was remarkably precise. They couldn't find any off-target edits to the DNA. And critically, it didn't cause the cell to blow past the natural, healthy stop codons that are supposed to be there. The cell's overall functions just weren't disrupted.
The potential here is just vast. One single optimized PERT therapy could, in theory, treat the specific nonsense mutations that cause diseases you've heard of, like cystic fibrosis, Duchenne muscular dystrophy, Batten disease, and literally thousands more. It's one drug for an entire class of genetic typos. And so that leaves us with this final massive question. Are we standing on the edge of a new era in medicine where a single one-time treatment really could cure thousands of different genetic diseases? Based on this research, the answer might just be yes.